This is an ultra-performance liquid chromatography, UPLC for short. UPLC is a powerful analytical instrument that we use to identify various compounds when designing new formulations or analyzing synthesized and biological samples. By setting up different mobile phase solvent systems, we are able to quantify drugs, lipids, or peptides, which is very useful in formulation development and assay validation. It comes with different modules such as PDA and mass spectrometry, which allows us to separate and detect compounds in complex compositions when designing methods specific to target analytes. Overall, this is a capable instrument we use on a regular basis in the Li Lab for drug delivery research.
Alright, so this is TFF system, which is tangential flow filtration. So we use it to either dialyze the particles, the nanoparticles, or concentrate them. So what we have here is a filter, and we can put our nanoparticles in here. So the particles will go into the filter. If it's in the, if you want to dialyze it, then we can add a buffer here, and then it can run in. The buffer, the used buffer can go into the waste. The new buffer will keep running. That's how we dialyze it. If we want to concentrate the particles, same thing, but we don't add to the buffer. So the particles will keep running inside the filter because their size is bigger than the pore size of the filter, and the buffer will go to the waste, and that's how we concentrate the particles, so the volume will get smaller, and the extra buffer will go to the waste.
This one is the rotavator. Usually we're using this rotavator to evaporate the organic phase in our drug solution. Like if we prepare lipsome, we need to have a thin film. So we dissolve our lipid in the organic phase, and then we're using this rotavator to evaporate those organic solvents so that we can get rid out of those like toxic organic solvent. And then after that we do the hydration to get our like nanoparticles. We label our drug with this red signal, so the computer can capture the red signal, and the blue signal is the nuclear. So when we put our sample in the confocal, this signal can be captured by the confocal, so we know how this drug distributes inside cell. In this case, they do not overlay with the nucleus. And we can also separate the different channels so we can see it more clearly. Now here is the nuclear, and here is the drug distribution inside cells. And another case, we can also using this confocal to do live cell image. In live cell image, we can have a vivid view of how the drug dynamic distribution inside cells. Also, in confocal, we can using confocal to see how our nanoparticle penetrated in different like cell model. In this case, we're using the spheroid model. It is multiple cell layers, and so by using confocal, we can know how the green signal now is our nanoparticle. We can see how this nanoparticle distributes into the different layer, because we have a spheroid. The spheroid, and under this confocal we can scan different layer from the surface to different depths of this spheroid, so that we can know exactly how much deep our nanoparticle can reach.